Welcome to today's lesson. Today we are dealing with 2018 biology practical. Let's enter inside and see what it contains. In question 1, the photographs below represent 3 mammalian bones labeled E, F and G. Should be able to understand and to know the drawings of the various mammalian bones. In this question, they are not very visible, but we will still answer them. E was the scapula, F is the humerus, and G is the ulna and radius. Now you are told, with a reason, identify the bones. Bone E, you have said it's a scapula. Reason is because has glenoid cavity. This is the glenoid cavity. This one, the glenoid is the glenoid cavity. Yeah, that's the reason it has the glenoid cavity. Also has the acromion. This is the acromion. This one, acromion, and this is the meta acromion. Yes, this is the spine. Yeah, you should be able to know those parts. Those reasons are okay. In F is the humerus. F is the humerus because, as you can see, though it is not very visible, has a rounded head. Has a rounded head. But in E, the scapula, you should be able to know where it is found. It is found at the shoulder, here up at the shoulder, and the humerus, it's below the shoulder. The shoulder has the reason is has a rounded head that articulate with the glenoid cavity. Now this round-headed head is here, it articulates with the glenoid cavity. Now here, those two are from the arm. Another reason is has condyles. The condyles articulates with the radius and ulna. And G now is the ulna and radius. Ulna has, the reason is because they have a sigmoid notch. This is the sigmoid notch, this hole, the sigmoid notch. This is the ulna and the radius. Yeah, ulna and radius has the sigmoid notch. The reason is has sigmoid notch. Also they have the greater tuberosity and the biceptal groove. You should be able to know the drawings of the mammalian bone. But a question in this here, it seems that they can bring the bones, so students should be able to know all types and drawings. You should be able to relate various drawings and know how to label them. In B, you are told, name the joints formed at the anterior and the posterior ends of F. F is the humerus. This is the anterior anterior, and the posterior. Posterior is the lower end. Here is up. This is lower. This is upper. Anterior and posterior. You should be able to know those terms. Anterior and posterior. Now, the joint in the anterior, the upper one, it is ball and socket joint. Ball and socket joint. And in the posterior, the lower end, is the hinge joint. They articulate there with the ulna and radius, the hinge joint. In C, state the types of movement facilitated by the joint at the anterior end of the specimen labeled F. We have seen the joint in the anterior end is the ball and socket joint. This joint, it rotates 360 degrees. So the movement is rotation of the arm. It rotates. Movement is rotation of the arm. It rotates in all directions, all planes. Rotation of the arm. D, Roman 1. Name the substance found inside the living tissue of the specimen represented in photograph M. Photograph M, we have said, is humerus. Now, in humerus, we found that it contains the bone marrow. Bone marrow, its function is helps in the manufacture of the red blood cells. Even we have been asked, state the function of the substance named. Help in the manufacture of red blood cells. C, Roman 1. Name the muscle bundle usually attached onto the front of the specimen represented into the photograph M. You are asked the muscle bundle of the humerus. You know that the muscle bundle in front is the bicep, while the muscle bundle that it is at the back, it is the triceps. Now you have been asked the front one, which is the biceps. Biceps. Spelling, you should be able to know the spelling. Biceps. State the function of the muscle bundle. The function is it relax and contract to move the lower arm. Contract or relax to move the lower arm. Question 2. Below is a photograph of the blood cells smear from a normal individual. The arrangement is arbitrary and the number of blood element is greater than that would normally occur in an actual microscope field. Although they are not very visible, but a student should be able to know the drawings of all the blood vessels. Like example, in red blood cell, you should be able to know they are biconcave in shape. In white blood cell, you should be able to know that they engulf themselves. They engulf or surround. They have the amoebic movement. Now, J, name the blood elements labeled J, K, and L. J we see is very small. It is very small, but it is a cone shape. It is a biconcave in shape. Now it is the red blood cells or the erythrocytes. And then K, as you can see, they have the amoebic movement. They resemble amoebic movement. Automatically, this is the leukocytes. cytes or the white blood cells. L, they are very small. L, they are very small, as you can see, but they are the thrombocytes. Thrombocytes or the platelets. State one function of each element named in A Roman 1 above. The function of the red blood cell help in the transportation. These help in transportation of oxygen and carbon dioxide. And K, which is the white blood cell, they protect by engulfing. To engulf is to surround, engulfing pathogens. These are the microorganisms causing diseases. Pathogens. L helps in blood clotting, which is the platelets help in blood clotting process. Yeah. In B, the photograph below is a section of a human intestine of a patient suffering from a common parasitic disease. Even though it is not very visible, this is a disease that occurs in a stomach. Now, there are very many diseases, but these ones are caused by parasites. Now, the disease is amoebic dysentery. This is the amoebic dysentery. It occurs in the stomach. Name the parasite that causes the disease. This simply are asked the causative agent. Now you know that the causative agent is Entamoeba histolytica. You should know this is a scientific name, so you should follow the rules, writing the scientific name by underlining them separately and starting with the generic name. You should start with the capital letter, with the species name or specific name with small letter. State 2 control measures for the disease. You should boil water before drinking. Boiling of water before drinking. Also, washing hands or fruits. Washing fruits or hands before eating. To prevent the amoebic dysentery. State 2 effects of having the part labeled G in patient's intestine. Part labeled G, these are the wounds, the stomach. The wounds, the effect is that you'll have pain, especially during feeding. You'll have pain especially during feeding. You have pain. Also, there will be blood stool. Even impaired absorption of digested food material. There will be impaired digestion or absorption of digested food material.
I hope you are enjoying the lesson. Do me a favor and subscribe. Thank you.
In question 3, you are provided with specimen labeled H. With the aid of hand lens, examine the external feature of the specimen. The specimen H was the bean pod. A pod from a bean. H was a bean pod. Bean pod. Now you are required to examine using the hand lens the features. A, you have been asked, what part of a plant is specimen H? Now the bean pod. You say this is a fruit. This is a fruit. Give two reasons for your answer. Why is it a fruit? Because it has two scars. That's the first reason you should give if you have been, as if you have written that your answer is fruit. Has two scars. That is the point of attachment to the receptacle. Has two scars. There is presence of seeds. Now presence of seeds and etc. Now you have been told open up specimen H longitudinally. Longitudinally is, you know how longitudinally looks like. Use a hand lens to observe the internal structures of specimen H. Yeah. Now you are being told draw and label the internal cut surface and associated structures of specimen H. Now you are required to draw well-labeled diagram of the bean pod which you have opened it. I should be able to know the drawing. Next, explain how you will determine the magnification of drawing made in B Roman 1 above. Now you should be able to know linear magnification. That is when you are using hand lens. Linear magnification is equal to the length of drawing all over the actual length of the object. Actual length. That is the linear magnification. Roman 3. State the mode of dispersal for seeds of specimen H. What the mode of dispersal? This is simply the self-dispersal mechanisms of the self-explosive mechanism. Self-explosive mechanism. Self-explosive mechanism. Explain how seeds of specimen H are dispersed through the mode stated in B Roman 1 above. The dry pods, due to water loss from the pod and pressure within the pod opens through the lines of weakness. Then the seeds are thrown away. Now you should say the dry pods loses water and a pressure is created, within the pod. Then the pod opens violently along lines of weakness. Then the seeds are thrown away, or they are dispersed from the parent plant. Thank you. Now welcome to today's lesson. Today we are dealing with paper 3 2022 March. Let's inside and see what it contains. In this year, question 1, they were provided with leaf E, F, G, H, and J, and they were required to construct the dichotomous key. Leaf E was a tricolor. And leaf F was maize leaf, and leaf H was a lantana, and leaf H was a twig, and leaf J was a jacaranda. Now you are required to construct a dichotomous key based on the following characteristics in order they are given. Now we start with leaf form. You start with number 1A, leaf simple, and leaf compound. Leaf simple, there were many, so you go to 2, while leaf compound was only 1, which was the jacaranda, which is J. In 2, leaf venation, leaf network veins, leaf with network veins, leaf with parallel veins. There were many with network veins, so we go to 3. While leaf with parallel veins was only 1, which is a maize leaf, F, specimen F. 3, according to leaf color, was a leaf with green color, and B, leaf with green color. You go to, there were 2, so go to 4, while leaf with green color was only 1, which was specimen E. Now we are left with G and H. They had their characteristics. Now the G had serrated margin, leaf with serrated margin, that was G. And B, leaf with smooth margin, that was H. And you have constructed a dichotomous key. In B, you can account for the likely observation if fresh specimen E was exposed to light and tested for starch. The specimen E was a tricolor. Now, it was tested for starch. The green part will turn blue-black with iodine solution. The green part will turn blue-black with iodine solution due to the presence of starch. Due to the presence of starch since photosynthesis occurred. Since photosynthesis occurred. If the part remained brown, therefore there was absence of starch and photosynthesis did not occur. In C, explain one observable feature that adapts plant from which specimen G and H were obtained to a dry environment. G had a hairy leafy surface. G have a hairy leaf surface. Also had its leaves were folding. Has folding leaves. This too was to reduce transpiration. Also the hairy leaf surface is to reduce transpiration. H had a shiny leaf surface or glossy. Had glossy leaf surface, also to reduce transpiration or water loss. To reduce transpiration, or you can write to reduce water loss. Even its leaves were folding, also to reduce the water loss. Leaves are folding to reduce water loss or transpiration. Yeah. In D, beside leaf characteristics, state one other observable characteristics on the plant from which specimen F was obtained that enable it to be placed in its class. F was the maize leaf. Therefore, you should be able to know the characteristics of class Monocotyledonine. First, has or have fibrous root system. Also have one cotyledon, and etc. You should be able to know the characteristics of class Monocotyledonine. In question 2, you are provided with solution M, which is the food substance. Here you are provided with reagents iodine, DCPIP, CPIP, and a filter paper, and a source of light, source of heat. You are required to test for food. Now you should be able to know the reagents is for testing or reagent provided. Let's start. In food test 1, let's test for starch because of iodine solution. Now the procedure will be to about 2 ml of food substance in a test tube. Add 2 drops of iodine solution and shake. Here the observation will be The brown color of iodine solution is retained. Now automatically starch is absent. Starch, conclusion, is absent. B, let's test for vitamin C. Vitamin C or ascorbic acid. Now here, this is the only unique procedure. You start with the reagent. You say to about 2 ml of DCPIP in a test tube, add the food substance. Dropwise as you shake. The observation will be DCIP color is decolorized, or DCPIP is decolorized. Therefore, vitamin C present. Vitamin C present. The last one, when you see a filter paper and a source of light, you are testing for lipids. Therefore, the food substance is lipids. Now the procedure will be put a drop of the food substance. Put a drop of the food substance onto the filter paper. Allow the drop to dry in the filter paper. Then hold the filter paper against a source of light. The observation will be not translucent or not transparent. No translucent mark is left on the filter paper. Therefore, the lipids were absent. When there is a mark, the lipids are present. In B, you are asked, state the precaution one should observe while conducting the experiment in 2A. You should avoid contamination of reagents. Also, avoid burning of the filter paper. Should avoid burning of the filter paper. Also, avoid misusing the reagents. Reagents, or also avoid misusing the food substance. Also, use clean apparatus. apparatus. Those are the precautions you should conduct when conducting the experiment. Question 3 and the last one. You are provided with specimen N and P. Here the specimen N was a leaf with yellow color, and P had green color. Therefore, you are told state observable difference between the two specimens, specimen N and P. The observable differences were: N had yellow leaves, while P had green leaves. N had thin stem, while P had big stem. Also, N had long stem, while P had short stem. So the features that were observed. Also, N had small leaves, while P had large leaves. In B Roman 1, name the phenomenon observed in specimen N. N is that it had yellow leaves. Now the phenomenon is etiolation. Etiolation is the phenomenon in N, the yellow leaves. Explain how the knowledge of the phenomenon named in B Roman 1 is applied in agriculture. Etiolation is applied in agriculture in many ways, but the main is when using a transparent polythene on a greenhouse, to enable adequate penetration of light for the crop. C. Account for the appearance of specimen N. The specimen had yellow leaves, or lacks chlorophyll because they were grown in darkness, hence couldn't carry out photosynthesis. In D, state 2 other environmental factors necessary for seed germination apart from light. Oxygen. These are the conditions necessary for germination. Oxygen, water or moisture, warmth, or optimum temperature. Lastly, E. State 2 observable features on the specimen that make them to be placed in the same class. Had broad leaves, taproot system, network veins, broad leaves or lamina. In short, these specimens are from class Dicotyledonei. Now the paper is over. Thank you.